We have a few more questions that we need to deal with regards to mitosis, and then we're going to transition into spending the last portion of our time together working through study guide questions that you may have in preparation for Monday's exam. All right? So how do chromosomes move along the spindle? Part of this we've already talked about, right? How do chromosomes move along the spindle? The motor proteins, right? The same way anything moves along a microtubule, it is carried along a microtubule by a motor protein. Okay? Now, just for added information, motor proteins that do not attach to microtubules on both sides are called kinesin. So kinesin is a motor protein that attaches to a microtubule on one side and something else on the other, either a vesicle or maybe a chromosome. All right, and then dynein are motor proteins that connect to microtubules on both sides. Yeah, kinesin. I'll write it down if I can get the lid off. So kinesin and then dynein. And so this would be microtubule on one side plus something else. And this is a microtubule on both sides. Now, what's the motor protein that works with microfilaments? It's neither of these. So a microfilament, the main one is actin. And what's the motor protein? Myosin. Good. All right, so how do chromosomes move along the spindle? Well, here you have a kinetochore microtubule. It is attached to the chromosome, not directly, but indirectly through a motor protein and then this kinetochore complex. And the motor protein will walk it to the end of the kinetochore microtubule. And then what happens once it gets to the end? It stops, yeah. When it gets to the end, it stops. And what happens to the microtubule? It starts shortening. So metaphase is very short, right? Metaphase is once it gets right to the end, and then you start anaphase, and it starts shortening and having each sister chromatid pulled to opposite ends of the cell. And so here's a research project that was done to figure out, are these microtubules shortening here, where what structure is sitting here? I know it says pole, but what structure is sitting there? Centrioles, right? Two of them forming the centrosome. So they want to know, is it shortening here or is it shortening here? Because if it's shortening here, that's a little weird because there's a motor protein attached to the very end of the microtubule. So nobody thought it was shortening here. So they irradiated it, lightening the color of the microtubules, and found out that it's shortening here at the microtubules. So what happens is a motor protein lets go, the microtubule shortens, it grabs back on, lets go, it shortens, it grabs back on, and it keeps doing that all the way until it ends up at the opposite pole where the centrosome is. And so here is that demonstrated. Here's the motor protein that walked it all the way to the end of the kinetochore microtubule. And then it has to let go and grab back on because it can't walk this way, right? The motor proteins only walk in one direction. So it has to let go. This shortens a little bit and it grabs back on. And it lets go and it shortens and it grabs back on. At least that's how we think it works. But nobody knows for sure because you can't see in that level of detail. That's how we think it works. All right? Any questions there? I know we talked a little bit about chromosomes moving along the microtubules already. All right. So here, let's sum this up. Motor proteins at the poles pull the kinetochore microtubules. So as they're shortening, there are motor proteins that are like reeling them in. On top of that, motor proteins near the kinetochore take the microtubule to the end and then pull them along the shortening microtubule. And then motor proteins on overlapping microtubules, the non-kinetochore, walk in opposite directions, right? Forcing those non-kinetochore microtubules in opposite directions, stretching the cell up. Stretching the cell, making it to where the poles are further apart. So it's easier to ensure that the two genetic copies end up on opposite ends of the cell and therefore end up in separate nuclei. Right? So that the cell can pass that G2M checkpoint. Okay, which checks to ensure that the copying was done correctly and that it was done properly, and then ultimately so that it can pass, the mitotic checkpoint, that things were attached correctly, separated correctly. All right? Okay, next question. What internal controls does the cell have over the cell cycle? And again, this is one we've already talked a little bit about. There are three main checkpoints. What are they? G1S, and what's the cell checking for there? So that's before the DNA is duplicated, right? So you're checking to make sure that the DNA is okay, is ready to duplicate, and you're mainly checking that you have everything you need in order to progress through S phase. You have all the energetic requirements that you need. You have growth factors to accomplish everything afterwards. Then we have what other checkpoints? So that's G1S. G2M, and what does the cell check for there? Was the DNA copied correctly? And are you ready to separate it into two nuclei? Right? Because you want those two nuclei to be genetically identical. Why do you want those two nuclei to be genetically identical? What are you trying to accomplish through mitosis? Yeah, you're trying to accomplish growth of a multicellular organism. You're not reproducing, you're growing. And so you need those cells to be genetic copies so that they both represent the organism correctly. And then what's our third checkpoint? The M checkpoint, the mitotic spindle, which is checking what? Right, if the chromosomes are attached correctly, both sister chromatids are attached to a kinetochore microtubule so that they can be switched properly. And so here is your various steps in the cell cycle. Here is S and G1, and then, so what was cool here is we had a cell that was stuck in G1. Okay, so this cell was stuck in G1. It's some cell, you might even call it G0 because it's left the cell cycle. And then it was brought into proximity with a cell that was already at S phase. And being brought close to this cell triggered this cell to actually transition over to S phase, to move past that G1S checkpoint. And this was one of the first experiments done to demonstrate that the cell needs to have something in place in order to pass through that checkpoint. So it's not just like the checkpoint is like a barrier that it has to overcome. The checkpoint is like a cost that has to be paid. Okay, there has to be something there to pay the price to move through that checkpoint. Okay, it's not just an obstacle. It's an obstacle that requires a cost, and not just a cost associated with the next phase, which would be S, which is a steep cost, but even a cost just to make that transition. Okay, like paying a toll. I just thought of that. It's a good analogy. Should use that one. Same thing is true for if you take a cell. This cell's at G1, and it was brought in close proximity to a cell that was mitotic and skipped entirely to M stage without actually going through S phase and G2, which means if this goes through mitosis, it's starting with half the amount of material, and that showed here, right? These chromosomes have two sister chromatids. These chromosomes do not, right? But again, this is demonstrating that there's something chemically that a cell has to express in order to move from one stage to the next. And so this cell here was transmitting or transferring whatever that chemical requirement was over to this cell and triggering it to move into the same stage that this cell's at. And so this got people thinking, what is that? What does this cell need to have in place in order to make that transition? And from Wednesday's quiz, what is it? So growth factors are external. So this is something that's internal, but it can come from somewhere else. And that's what's happening here, is it's coming from somewhere else, but it's actually an internal component. It has to be transferred inside of the cell. Cyclin. You got to speak up so I can hear you. I'm old. And so here are our 3 checkpoints illustrated on our cell cycle. Here's our G1S checkpoint. Here's our G2M checkpoint. And here's our M, or mitotic spindle checkpoint. And in all 3 of these, again, it's more than just some obstacle that's checking for make sure everything's right, and then you can progress. The cell also has to make something chemically to allow it to pass through that. And what it's making chemically is a specific type of cyclin. So you have different types of cyclin for every single one of these checkpoints. And that's how you were able to, in this one, induce this cell to move all the way from G1 to M, because what this transferred over was the cyclin that allows it to move past this G2M checkpoint, which is this cyclin, or is the degradation of this M cyclin. After the M cyclin's there, moving into G2, and then its degradation. Okay, so there's a specific cyclin associated with all of these. Now, CDK, do you remember what this stands for? Cyclin-dependent kinase. And we've seen kinase before. We saw kinase in glycolysis, and a couple of our enzymes end in a kinase. And what does a kinase enzyme do? It phosphorylates other enzymes. And phosphate groups turn on or turn off enzymes. So a kinase is an enzyme that activates another enzyme. And then this kinase, this is called cyclin-dependent kinase, meaning the kinase can only do its work in the presence of what? What it's dependent on, which is cyclin. So if you have the right cyclin to activate this enzyme, then this enzyme can phosphorylate other enzymes, turning them on, allowing the cell to progress past that checkpoint. So everything is not just a check for quality, but it's also a check for the right chemical requirements. And then that way, even though a cell may have what it needs to go through that step, if it doesn't also have the chemical requirements to move through it, it won't go through it. So it's a way a cell can keep from moving through the cell cycle, even if it has the energetic requirements to do S phase. If it's not producing this G1S cyclin that would activate this and would cause the shift from G1 to S. Yeah, Kinsey.
So would it not have a cyclin like if it didn't want to grow anymore, or like when to stop it?
Right. Yeah, so cyclin is a protein that would be transcribed and then translated. And then so if it doesn't want to grow anymore, it will stop producing that protein. Yep, so the transcription of that gene for that G1S cyclin would stop. Yep.
And then it can start back up. Like
Sure, yeah, absolutely. And this is one of the ways in which we're trying to trigger neurons to re-enter the cell cycle and to divide, is by getting them to actually synthesize the G1S cyclin when they normally wouldn't. They've left the cell cycle. All right? Does that make sense? Okay. So there's a couple of summary slides summing up this work, but we've already talked through this. So I will leave these to you. It's just going over what we've already gone over. But I want to, again, give us some time at the end of class to work through study guide questions. All right? So how does the organism maintain subpopulations of cells? How does the organism maintain subpopulations of cells? First of all, what is a subpopulation of cell? Yeah, not even of just a different tissue necessarily, but a cell of a different type even within the same tissue. Right? But yeah, I mean, obviously that's going to have an impact on developing different tissue types. But this is a way in which an organism, if it's going to control division and keep cells from going through mitosis, it needs to do that in such a way that it still maintains a certain amount of whatever that cell type is. Okay, so we need to talk through this. I don't have any pictures that go along with this, and so we'll talk about this. So internal controls, the production of those cyclins, the production of G1S cyclin or of M cyclin, all of that can be controlled by external factors, by hormones and growth factors, hormones that can slow or speed up or even stop the production of certain proteins inside of the cell. And so these are external controls that then have internal consequences. External controls that then have internal consequences. This is one of the hallmarks of being a multicellular organism. How would you describe what's going on here? If you were to use one word to represent what's happening here with external controls regulating internal features, what word would you use?
Complex.
Okay, we've got a vote for complex. Any other ideas? What's that? Design. I like that one. Any other ideas of a single word to represent what's going on here? Of having external controls impacting internal events. Okay? Communication, I like that one too. And it's a really sophisticated communication. I know that's two words. It's a really sophisticated communication. I like the term regulation as well. And all of this is kind of a hallmark of a multicellular organism, because an individual cell does not a multicellular organism make, right? Right? A single cell doesn't make a multicellular organism. And so there are two hallmarks of multicellularity. One is differentiation, or if you call it division of labor, where you actually have different cells carrying on different tasks. And the other one is a great deal of regulation, or communication, or design. Okay. Those are the two hallmarks of multicellularity. So cells physically touching each other, these are trigger contact inhibition, and they actually shunt out of G1 into G0 or G0 by contact inhibition. Any loss in cell cycle control gives rise to uncontrollable growth, which is some kind of a tumor, and then if the cells lose their ability to remain adhered together, in addition to losing their ability to control their cell cycle, then they metastasize and they move, and that would be the transition from a benign tumor to a cancerous tumor. So they lose contact inhibition. They lose any possibility of being inhibited in the future, because they lost whatever left them attached to other cells. So this is usually caused by mutations in genes. We call these oncogenes, genes that mutations in them lead to uncontrollable growth. And it usually is a gene in this cyclin and cyclodependent kinase complexes. Some kind of a mutation there that either leads to a constant production of that G1S cyclin, so the cell constantly goes through there even if it's being told not to, because it's got no way to regulate that, or constant production of a mutated form of the cyclodependent kinase that stays active even if cyclin isn't present. Okay. Make sense? Okay.
Is there any kind that completely gets rid of it?
That completely gets rid of?
The gene?
That completely eliminates the gene altogether?
Yeah.
Well, if you couldn't make the cyclin at all and your cyclin-dependent kinase was fine and can't operate in the absence of cyclin, what you would have is just a cell that couldn't go into the cell cycle. And that wouldn't necessarily be a bad thing. That cell would just probably either die on its own or would go through apoptosis and be replaced. Yeah,
So it
Charles?
sounds like cancer is a genetic thing where it can't produce those different regulators.
Yes, but it doesn't have to be an inherited genetic issue, right? It could be a genetic issue that happens as a result of a genetic change within that generation.
How does that affect, is it even possible to find a cure for cancer if it's so...
Well, you'd have to find a cure for every individual cancer, because every cancer is different. Yeah, every cancer has a different mechanism behind it. Therefore, every cancer has its own cure. Now, there are treatments that work for varieties of cancer, because there are certain things that tend to be true of all cancerous cells, and so you can target those aspects. Like you can target cells that are free-floating, right? Because that's not normal for a cell, except for specific cell types that have specific markers. So there are ways that you can just approach all forms of cancer, but typically every form of cancer would require a separate cure. And then every cancer, even the same cancer, is different in every individual because the cells are different genetically. Yeah.
The difference between benign and non-benign?
The difference between benign and metastatic or cancerous is the physical adhesion of the cells to the growth. And so in a cancerous growth, the cells are no longer adhered and they're growing out of control. And so they're what's called metastatic. They move. They enter into the bloodstream and they move. They migrate to different tissues. Usually they migrate through the lymphatic ducts. The lymphatic ducts is basically the sewage system of the body. And so that's usually how they migrate. They get into the lymphatic ducts and move from there into the bloodstream and then end up in various places. My son's down here, by the way, if you're wondering why he's hiding under the podium. So. What's that?
Sounded like a joke.
It is not a joke. No, my son is really down here hiding under the podium. So. All right, so how does binary fission differ from mitosis and cytokinesis? How does binary fission differ from mitosis and cytokinesis? And the reason why I wanted to address this question is I wanted to further remind you that mitosis is not cell division, right? Mitosis is nuclear division. Cytokinesis is cell division. All right, so one way to answer this question, how does binary fission differ from mitosis and cytokinesis, is binary fission is a single process. Mitosis and cytokinesis are two processes, right? It's like binary fission is one process, and then mitosis and cytokinesis, two separate processes. So that's one way in which you can answer this question. And then there's some other ways illustrated in this diagram. So looking at this is binary fission of a bacterial cell. Take a look at this. Just chew on it for a little bit and see if you can see some differences between this and what we've talked about in mitosis and cytokinesis. Whenever you're done chewing, feel free to share your thoughts. If you want to say something, Alyssa?
Well, it doesn't have any mitosis at all.
Right, it doesn't have any mitosis. Well, I mean, what is true of mitosis that isn't true here?
There's no condensing. There's no lining on the plate.
Okay, so yeah, there's no nucleus, right? So you're not dissolving a nucleus. You see a mitotic spindle. There's no mitotic spindle. So then there's a question, how on earth does the chromosome get segregated into two different regions without a mitotic spindle? It's a wonderful question. It actually uses a different cytoskeletal element. It uses things that are similar to microfilaments to migrate it. Any other differences? Yeah, so you don't have metaphase, you don't have prometaphase, you don't have anaphase, you don't have telophase, right? Your chromosome isn't linear like it is in mitosis and eukaryotic cells. Instead, it's a single circular chromosome.
Isn't it forming a peptide-like wall each way?
Yes, yeah, but plant cells do that as well, right? They form that plate as they go through mitosis. Plant cells also don't have centrosomes like animal cells do.
So how would this process evolve into...
That's an excellent question. We'll talk about that in just a second. Yeah. And so as you look through here, one other big difference is replication of the chromosome happens along with the separation of the chromosome. That both are happening simultaneously. Okay? So that's another big difference between binary fission and mitosis and cytokinesis. So just to sum up some of these differences, chromosomes are rarely visible during binary fission. On that diagram, we showed you the chromosome, but if you were actually watching this under a microscope, you would not usually see the chromosomes, which in eukaryotic cells is not the case. You can very easily see the chromosomes during mitosis and cytokinesis. It's unclear exactly how the chromosomes segregate. It's certainly not with a mitotic spindle, but it still involves the cytoskeleton. Most prokaryotes have a single circular piece of DNA rather than several linear chromosomes. In prokaryotes, DNA replication fills most of the cell cycle and happens along with the division. And so, as an example, in E. coli, when they are growing during their exponential phase, replicate every 20 minutes. Their doubling time is 20 minutes, and it requires 19 minutes to replicate the DNA. So through most of the time while the cell is growing and getting ready to divide, the DNA is being replicated at the same time. And then the last question that we need to deal with, which Charles just asked, how does mitosis develop from binary fission? It's what we're going to deal with in this question. Where did mitosis originate? How did it develop from binary fission? It's an excellent question. All right. So yeah, the idea here is that mitosis evolved from binary fission as a requirement after the development of the nucleus. Right? So one of the first differences y'all pointed out in between binary fission and mitosis and cytokinesis is no nucleus. Okay, so after the nucleus, you now have a requirement to do things a little bit differently. Now, what's interesting is you have some of our protist groups, some single-celled eukaryotic forms, that have a cell division that has some pieces of mitosis and cytokinesis, but also lacks some of those characteristics. So dinoflagellates probably use microtubules, although as far as I know, nobody's actually demonstrated microtubules segregating the chromosomes in a dinoflagellate. Instead, their chromosomes are attached to the plasma membrane, which is what happens during binary fission, that the bacterial chromosome is physically attached to the membrane of the bacterium. Yeast, which is a type of fungal cell, and diatoms, the nucleus stays together during the entire process. They still use a mitotic spindle, but the spindle develops inside of the nucleus. The nucleus doesn't dissolve during prophase, and so prophase, the chromosomes still condense. Your centrosomes migrate to opposite sides, but not to opposite sides of the cell, to opposite sides of the nucleus. And then they stretch the nucleus out and pinch that into two when it's time.
So that's not considered mitosis. It's still a eukaryote.
It is, but it's not like mitosis in animal cells. Yeah. It's something in between what bacteria do and what animal cells do. And then plants, they don't have centrosomes. They still produce a mitotic spindle. The nucleus still breaks down like it does in animal cells, but they don't have a centrosome. And so then the question is, how do you get mitosis from binary fission? And the answer to that question is nobody really knows. Like, we don't really have a good mechanism. We do have some organisms that do different forms of mitosis, but there's not really a good mechanism for getting mitosis from binary fission. And so I don't know if I've already told you this or not. I'm sure it may have come up in a previous discussion, but an idea that's actually gaining in popularity is that the original organisms, the original survival machine was actually a eukaryotic cell, and that prokaryotic cells are derived from that the original life form is actually a eukaryotic cell that already had a nucleus. And I think that idea would make this easier to explain. Be easier to generate binary fission from mitosis. You just lose some of the mechanisms than to go the other way. What are some of the machines that you would need to develop in order to move from binary fission to mitosis? What are some of the machines that are involved in mitosis that you have to generate?
Histones?
What's that?
Histones?
Well, so that's not part of mitosis, but yeah, you'd also need to develop a way to organize a lot more DNA, and that's where histones come in. Centrosomes, right? Although plants do it without centrosomes, but you need some kind of microtubule organization center so you can develop a mitotic spindle. Although dinoflagellates don't use a mitotic spindle and use something else, but you would need to at least do that to get to animals. What else? What's that? Motor proteins to walk along those microtubules. Good, I love it. Something to dissolve the nucleus. Yeah.
Structural change from the cell wall being the cell wall to now being a membrane.
Yeah, oh, absolutely. And not only that, you would need to develop a way by which the chromosome no longer is physically attached to that membrane, right? Regulatory enzymes, right? You need all those cyclin-dependent kinases. You need genes that are going to make the specific cyclin for all of those transitions. Talking about a lot of machinery that needs to develop in order to move from binary fission to mitosis and cytokinesis. It's not a simple transition. But if it went the other way, this is a simpler process, right? Talking about losing some of those machines. All right? So how do we get mitosis? I don't know. Unless your view of origins doesn't require rooting all life back to an original survival machine, then it's not an issue. right? Then the reason why different eukaryotes have different forms of cell division is not because it's better or worse, it's just because that's how they were designed. And so it's something that is maintained through time.
So is
Yeah.
mitosis one of the reasons why some people are thinking that eukaryotes came first? And then
It's one of the
prokaryotes?
reasons, yeah. It's one of the reasons why. Another reason is we really don't have a good mechanism by which